Good morning, students. Now coming to structure of embryo, dicotyledonous embryo and monocotyledonous embryo. Students, after fertilization, zygote is formed as a result of fertilization. This is 2A. So, in case of higher plants like angiosperms, where the fertilization takes place in female parent, it is takes place in a embryo sac. Zygote is formed in embryo sac. Similarly, this embryo sac zygote is situated towards the micropylar end. Micropylar end. Micropylar end of embryo sac. Because towards the micropylar end, embryo sac contains egg apparatus, and egg is present at the central part of egg apparatus. So zygote is formed towards the micropylar end inside the embryo sac. So once this zygote is formed as a result of fertilization, now the zygote it undergoes mitosis, leading to the formation of embryo. So zygote is 2N. So embryo is also 2N. Such formation of embryo from zygote as a result of mitosis, this is called embryogeny. Embryogeny, also called embryogenesis. Embryogenesis. So the formation of embryo from zygote is called embryogeny or embryogenesis. The process of formation of embryo from zygote is called embryogeny or embryogenesis. So the zygote is develops into embryo only after a certain amount of endosperm is formed. So endosperm, endosperm, it is formed from primary endosperm nucleus. Endosperm is triploid, and endosperm is post-fertilization product in angiosperms. Endosperm is the food for developing embryo. So, endosperm formation it proceeds. Formation of endosperm it proceeds the formation of embryo. Why endosperm is formed before the formation of embryo? Because endosperm is the nutritive tissue for developing embryo. So only a certain amount of endosperm is formed. After a certain amount of endosperm is formed, now embryo development it starts after the formation of endosperm. Embryo development it takes place only after a certain amount of endosperm is formed, because endosperm is the nutritive tissue for developing embryo. So endosperm is formed first and then it proceeds in embryo development. Similarly, monopod seeds and dipod seeds, they show many differences. They are varying. Dipod seeds and monopod seeds are varying, but the initial formation of the initial development of embryo in dicots and monocots it is similar. Initial development very important. Initial development of embryo in monocots and dicots almost similar. So even though some differences are present between dicots and monocot seeds, development of embryo inside this dicot and monocot seeds initially it is similar. Now coming to the dicotyledonous embryo. Dicot's embryo, the structure of dicot's embryo. Every dicotyledonous embryo. Okay, first zygote it develops into proembryo. Proembryo. First the nucleus of that zygote divides by mitosis, leading to the formation of some amount of cells, that is pro-embryo, primary embryo. Now this pro-embryo, it develops into heart-shaped and globular and mature embryo. So first zygote develops into pro-embryo, primary embryo, immature embryo. Now this immature embryo subsequently it develops into the mature embryo. Mature embryo in dicotyledonous plants, this is heart-shaped, globular and it is mature embryo. In this way, every dicot embryo, it is represented by mainly two parts. One is embryonal axis, okay? One is embryonal axis and two cartilages. So this is dicotyledonous embryo. This embryo is represented by embryonal axis. This is embryonal axis. Then it is also represented by two cartilages. There are two cartilages. These two cartilages and this one is embryonal axis. In this way, mature heart-shaped globular embryo in dicots is represented by embryonal axis and two cartilages. Actually, these cartilages, there are two cartilages and these two cartilages, they are represented by reserve food material. Reserve food material. They store some amount of food material. Due to storage of food material inside this cartilages of dicot cell, it is very fleshy, very important. Cartilages in dicotyledonous plants, they are very fleshy. Why cartilages of dicotyledonous seeds, they are very fleshy? They store some amount of food material. In dicot seed, food materials are stored inside. Most of the dicotyledonous seeds, food material is stored inside the cartilages. So the cartilages, they become succulent and they become fleshy. So there are two cartilages and the two cartilages, they are the site of reserve food materials and they remain fleshy. In this way, two cartilages and one embryonal axis is there. Now coming to another point, very important. Here, this is embryonal axis. Okay. The next part of the dicot cell is embryonal axis. This embryonal axis, this is embryonal axis. The point of embryonal axis, that is equal to the level of cartilages. Okay. This point of embryonal axis. The point of embryonal axis present equal to the level of cartilages, that is called cartilaginary node. Okay, this is cartilaginary node. So what is cartilaginary node? The point of embryonal axis present almost equal to the level of cartilages. This is equal to the level of cartilages. Such point of embryonal axis is called cartilaginary node. This is called cartilaginary node. Okay. Similarly, the point of embryonal axis, this point, the point of embryonal axis present above the cartilaginary node. The point of embryonal axis present above the cartilaginary node. This is called epicotyle. This is epicotyle. Very important. What is epicotyle? The point of embryonal axis present above the cartilaginary node. This is called epicotyle. This is epicotyle. This epicotyle, it terminates into a small flamen. Okay, this epicotyle, it terminates into a small plumule. Plumule is nothing but the stem tip. Stem tip. Or this is also called the primary stem. This is the primary stem. So, plumule is nothing but the primary stem. In further developmental stages, this plumule develops into the stem, primary shoot. Plumule is the primary shoot. Plumule is the primary stem. Plumule is the primary stem tip. In this way, this epicotyle it terminates into a plumule consisting of that is the stem tip or primary stem. This is plumule, primary stem. Similarly, the point of embryonal axis present below the cotyledonary node. The point of embryonal axis present below the cotyledonary node. This is called hypocotyle. Very, very important. Hypocotyle. Hypo means lower. This is hypocotyle. Actually, this hypocotyle it terminates into the radical. Very, very important. This terminates into the radical. Radical means primary roots. This is primary roots. And this radical is also covered and protected by a thimble-like protecting sheath. You know this pointed root modifications and root regions. Okay? The thimble-like protecting sheath present surrounding this radical. This is called a root cap. This is a root cap. It protects the actually dividing meristematic cells present at the tip of the root system. In this way, the dicotyledonous plant, dicotyledonous embryo is represented by two cotyledons and embryonal axis. Cotyledons are very fleshy. They store some amount of food materials. That's why they become fleshy and succulent. Similarly, this embryonal axis consists of a point where it is equal to the level of cotyledons, that is called cotyledonary node. Point of embryonal axis above the cotyledonary node, this is called epicotyle. It terminates into plumule. Similarly, point of embryonal axis present below the cotyledonary node, this is called hypocotyle. It terminates into radical and which is covered by a thimble-like protecting sheath. This is called root cap. Students, this is the structure of dicotyledonous embryo. Now coming to monocotyledonous embryo. Okay. So, in case of monocotyledonous embryo, monocots, only one cotyledon is there. Only one cotyledon, and this also consists of embryonal axis. Embryonal axis. So, the main difference between dicot and monocot embryos, embryo of dicotyledonous plants represented by two cotyledons, and only one cotyledon is present in monocotyledonous embryo. Embryonal axis is common. Okay. So, in this, in case of grass family, grasses, that is a rice, wheat, sorghum, like that. The cotyledon present in monocotyledonous family, that is in grasses, shield shaped single cotyledon is present. Shield shaped single cotyledon present in grass family, that is called scutellum. This is scutellum. So, this is scutellum. Very, very important. Means this is cotyledon. Scutellum is nothing but single shield shaped cotyledon present in monocotyledonous plants like grasses. So, In case of this monocot embryo, only one shield shaped cartilage is present, that is called scutellum, and embryonal axis is also there. Okay, so here the single shield shaped cartilage is attached with the embryonal axis. The point of embryonal axis where the cartilage it is attached, such point of embryonal axis is called epiblast. The same point in dicotyledonous embryo, this is called cartilaginary node. The same point is called cartilaginary node in dicot embryo. But the embryonal axis where the cartilages are attached, the embryonal axis in monocot embryo where the single cartilage is attached, that is this location. This is called epiblast. Similarly, the point of embryonal axis, this point present above the attachment of embryo, attachment of cartilage, means the point of embryonal axis present above the epiblast means attachment of cartilage to the embryonal axis. Such a point is called epicotyle, just like this. Both are equal. This is called epicotyle. Very important. Epicotyle is present in dicot embryo and also monocot embryo. Epicotyle. Now this epicotyle it terminates into leaf primordia. Leaf primordia. And also it gives plumule. So leaf primordia means young leaves, very young leaves present in embryo. They are called primary leaves or leaf primordia. So the point of embryonal axis where the cartilage is attached is called epiblast. The point of embryonal axis present above the epiblast is called epicotyle. This epicotyle terminates into primary leaves, primordia and plumule. And these primordial leaves and plumule, they are covered by a protecting sheath. This is a red colored sheath. This protecting plumule and primordial leaves, they are completely protected by a sheath. Such a protecting sheath is called polyoctile. This is called polyoctile. This is the specialty of monocot embryo. This protecting sheath is completely absent in dicotyledonous embryo. Okay, plumule is covered by root cap only. There is no the special protecting sheath surrounding this plumule in case of dicot embryo. But this epicotyl gives primordial leaves and plumule in monocot same. These primordial leaves and also plumule, they are completely protected by a protecting sheath. Such a sheath is called coleoptile. Very important. At the same time, the point of embryonal axis, this point, this is the point of embryonal axis present below the epiblast. This is present below the epiblast. Epiblast. Such a point of embryonal axis present below the epiblast is called this is hypocotyle. This is hypocotyle. Just like in dicotyledonous root, hypocotyle, it is terminates into radical. Terminates into radical. But in monocotyledonous, its radical is temporary. Soon it is replaced by a group of fibrous roots. Initially, radical is formed in monocot seed also, but radical in monocotyledonous plants is soon replaced by a tough hair-like fibrous roots. So hypocotyl terminates into radical and also root cap. Radical and root cap. Just like here also the radical and root cap, they are completely protected by a sheath. This is called coleorhiza. Very very important. Coleorhiza. This is the protecting sheath of radical and root cap. Similarly, coleoptile is the protecting sheath present surrounding primordial leaves and plumule. Students, very very important. Dicots and monocots embryo. In dicot embryo, two cotyledons are there, but in monocot embryo, only one cotyledon is there. In dicots, food materials are stored in cotyledons. In monocots, food material is stored in endosperm. Single cotyledon is called scutellum. Epicotyl, hypocotyl both are present in both embryos. But cotyledonal node is replaced by epiblast in monocotyledonous embryo. And coleorhiza, they are present in monocot embryo but absent in dicot embryo. Very very important. Now coming to other points. Students, in case of some plants, okay, in case of some plants like beets and black pepper, beets and black pepper. In these two plants, okay, in these two plants, here remnant nucellus. Some amount of nucellus is left out inside the seeds. Actually, nucellus is the food material for developing embryo sac. As the embryo sac increases in its volume inside this ovule, completely nucellus is disappear. But some amount of nucellus is left out inside the seed even after the completion of the development of embryo sac. Such a remnant nucellus, okay, such a remnant nucellus present in the seed is called perisperm. This is called perisperm. Perisperm is nothing but nucellus, but it is present inside the seed. When this nucellus is present in ovule, now this is called nucellus. When some amount of nucellus is left out inside the seed after completion of fertilization, now this is called nucellus. Students, what is the ploidy? What is the ploidy of perisperm? Perisperm is nothing but nucellus. What is the ploidy of nucellus? Diploid. This is a diploid. So, perisperm is the remnant nucellus present in some dicotyledonous plants. That is in beets and black pepper. Remnant nucellus is called perisperm. Example, beet and black pepper. Similarly, in case of monocotyledonous plants, in monocot seed, like grasses, alluron layer is also there. Alluron layer. What is alluron layer? The covering, the external covering layer of endosperm that separates the endosperm from embryo. Very very important. Endosperm is there. The protecting, the layer, the proteinaceous layer present surrounding the endosperm. The proteinaceous layer surrounding the endosperm in monocot seed, that is called alluron layer. Alluron is equal to proteins. It is made up of proteins. But what is the ploidy of alluron layer? Alluron layer is nothing but nucellus, covering of nucellus. What is the ploidy of nucellus? What is the ploidy of endosperm? Angiosperms? Triploid. So alluron layer is made up of proteins and it is present surrounding the nucellus, surrounding the endosperm. So endosperm and alluron layer both are similar, and both are triploid in nature. This is the ploidy of alluron layer.